So without any further ado, David's going to take us down a different journey than we've heard him talk about so much in this forum years ago, and I look forward to his presentation. David.
Thanks everyone for coming, and it's great to see lots of familiar faces, and I've enjoyed a little bit of sunshine away from the Scottish winter. This is the beautiful Midlothian where the Roslin Institute sits just here. My office looks out over the Pentland Hills. This is the Morden Research Institute, Scotland's Agriculture College, Biocampus, not entirely filled, but this is the highest concentration of animal sciences in Europe. And really quite a stimulating place to be. And this is the Roslin Institute building, which we constructed and moved into in 2011. So what I'm going to talk about today is a little bit of various things, wandering through things that interest me. Firstly, talking a little bit about livestock genetics, which is what Roslin is actually famous for. Everyone thinks of Dolly the sheep, but in fact what Roslin is famous for is quantitative genetics. It actually owes its origins to the Institute of Genetics of the University of Edinburgh, which was the home of such luminaries as Waddington and Fisher, famous names in statistical genetics, and has continued to be a mainstay of the development of livestock quantitative genetics. So I'm going to talk a little bit about that there when genomics is changing the way we do genetics, and finally some of my own, and I have to say macrophages will get a look in, some of my own interests. So livestock agriculture is controversial. There are battle lines drawn. On the one hand, the opponents, mainly vegetarians I imagine, who are opposed to any form of livestock agriculture, and on the other hand, those who recognise that livestock agriculture is a critical part of agriculture. It's particularly important, and we've recently set up a collaboration with the International Livestock Research Institute in Nairobi, to the main route out of poverty for the poorest farmers. The poorest farmers, particularly women, start with chickens, then small ruminants, then dairy cattle, and that is a major source of both protein and nutrition for their children and funds to lift themselves out of poverty. So we have a large centre funded by the Gates Foundation in that space. But dealing with 2050 and everyone telling us there's going to be 9 billion people in the world, we also need intensification of production systems. We need to reduce waste and pollution. We need to increase the efficiency. We need alternatives to antimicrobials to improve disease resistance, and we need better food safety. So we really need to think about how we deal with the genetics of animals. What a lot of people don't know, and what I didn't know until I went to Roslin, is that there has been a massive increase in the productivity of livestock animals in the West, really through conventional breeding and more recently through genomic approaches to breeding. For example, the average layer hen between 1960s and 2005 increased the number of eggs that it produced per year and almost doubled the number of eggs produced per tonne of feed. If you do the arithmetic, in fact, the average layer converts almost half of the food that they eat into eggs. Now, that's an extraordinarily efficient animal. So once we went beyond conventional breeding, the Genomics Revolution, when we started getting high-density genetic markers, we started being able to, instead of breeding empirically, looking at the performance of the progeny and saying, oh, that's a good bull because his calves produce lots of milk, we started being able to assess the barcode. We weren't entirely predictive in the early days, so the outcomes were sometimes unpredictable. But in Western breeding, as particularly, for example, the Holstein Friesians of the United Kingdom, it's now possible to establish a genetic barcode. In fact, the costs have come down to the point where we can establish it for both the cow and the bull, instead of relying solely on the genotypes of the bulls for breeding. And you can simultaneously optimize that barcode for longevity, for udder health and resistance to mastitis, ease of calving, disease resistance for tuberculosis, which is a major issue in the UK. We've recently implemented genomic selection for tuberculosis resistance in the UK dairy cattle. And that's particularly good news for farmers and even better news for badgers. So all of these things, and particularly starting to look much more at whether we can breed for improved utilisation and reduced methane production. But beyond the point of choosing genotypes and selecting them, we're now in the era where we can look much more at selecting genotypes and modifying genotypes, and really the genetically modified animal is on our doorstep. We've used genetically modified animals for a very long time. We've optimized their production by heterosis, by deliberately using hybrid vigor. It's, of course, possible, and with increased technologies, it's possible to drive this to make hybrids of sheep and goats, of alpacas and llamas, of bison and cattle. That would obviously be a genetically modified animal if you chose to see it that way, but we don't have too much problem with it. I often give lectures on genetic modification, and I point out to people that if wheat was invented today, it would never be approved. It's a hybrid of 3 different species, one of which we don't know. It's been extensively genetically modified, and it causes allergies in humans, in celiacs. So there's no way that wheat would ever be approved. We have cloning, and this was actually obviously a technique pioneered by Roslin. They imported cloned animals into the United Kingdom, and there was a massive furore about the evils of cloned animals. There was a cartoon where the husband is holding a bottle to his wife and saying, "Does this milk smell cloned to you?" There is mutagenesis and selection. We're not really allowed to do that readily in animals. Of course, that's the mainstay of plant breeding. There is what we might call cisgenesis, where we move a desirable gene from a breed of the same species. For example, taking a desirable trait from a Charolais or a Texel and just moving that particular form of the gene into another animal of the same species to rapidly introgress. But this is what most people understand by genetic modification, and that is transgenesis. And transgenesis has become fairly controversial, but it's also quite useful. So we in Roslin have made transgenic chickens in which all of them are all over fluoro green. This one feels obviously a little left out. We have transgenic pigs. This is the front end. The transgenic males have a very interesting trait. The back end really glows green. And of course, Dr. Seuss was obviously prescient that green eggs and ham really is now possible. But in the world of real production, the Aquabounty salmon has now finally been approved 20 years after it was first made in the United States. We were afeared of releasing these, even though the only animals that are grown are sterile females, but quite relaxed about having these things brought in as pets into the United States, got into the waterways, and essentially they eat everything. They can survive for prolonged periods out of water, and they've almost obliterated the fish populations of the Great Lakes. So the threats of these versus introduced species are rather overblown. And the other one that actually died a death was the so-called enviro pig. Both chickens and pigs are unable to metabolize the phosphate in grain, and to do so they need the enzyme phytase. So the enviro pig had a transgene that expressed phytase specifically in salivary glands. That enabled it to much more effectively utilize the phosphate in grain, reduce the amount of phosphate coming out the other end, remove the requirement to supplement the feed with phosphate for both chickens and pigs. Within weeks of the release, this came out. A pig, this is one of the earliest examples, really, of a transgenic animal that got its own song within weeks. But following on from the creation of these animals, the genetic selection was applied, and you can see that the transgenic animals actually showed genetic gain faster than the non-transgenic animals. And so it was possible to actually breed to utilize that transgene more effectively and to improve its ability to both enable growth and reduce environmental impact. Unfortunately, these animals have been culled now, and there is no prospect of them being released into production. So transgenic chickens, we've made lots of them in Roslin, and this one is the transgene. This is a transgenic bird that is resistant to transmission of influenza. It was published a few years ago, and we're still working on transgenic approaches to reduce the impact of avian flu. I think that's probably the best chance. This animal is a gynandromorph. This is a perfectly natural animal. It's male on this side, male spur, male comb, female on this side, and that's rather interesting biology because it tells us that in birds, having a testis is not sufficient to make you a male. Every cell in the body of a male or female chicken knows that it's male or female. This is the famous Scottish dumpy, which has some interesting mutations affecting musculoskeletal development. These are various feather patterning variants that we've worked on in Roslin, which involve large translocations. And these are the sorts of birds that are quite popular in China that have a mutation in melanocyte differentiation. Of course, we can wait for those sorts of things, but the real revolution in livestock genetic variation is, of course, genome editing. This started with the talons, which basically use synthetic DNA binding protein coupled to the FOC1 nuclease to create double-stranded breaks in DNA, which can either be repaired by non-homologous end joining, or if you introduce a template, you can actually get homology-dependent repair. And in Roslin, we created the first genome-edited pig, not particularly imaginatively named Pig 26. There wasn't an obvious, it could have been called Miss Piggy, except it was male. And Pig 26 had a single base substitution, and we made this animal to make it resistant to African swine fever, having identified a single base or single amino acid difference between the African pigs, including warthogs, and the domestic pig, which we believe is responsible for their differential response to African swine fever virus. And these animals are currently being bred up and tested. So that was published a couple of years ago now in PNAS. Of course, everyone probably in this room has heard of CRISPR-Cas9. This has made the genome editing considerably easier. In fact, this slide I just took off a commercial website which will provide you with the guides and the Cas9 put into a plasmid for you. I have a PhD student in my lab who came from Mexico with almost no molecular biology experience, created the vectors within a couple of weeks and knocked out my favourite enhancer that I've been working on for 20 years in a matter of a couple of months. It's really quite scary. So if we have all that technology and we're starting down the road of genetic manipulation, the question is, what should we edit? And that's where the other major revolution comes in, and that's the genomic revolution. Roslin, the University of Edinburgh, in fact, already had one of the largest sequencing facilities in the UK, and recently we installed the Illumina X10 sequencing platform, which enables us to sequence 20,000 human genomes a year. That has been released for non-human genomes at high density, so we are currently sequencing literally tens of thousands of chicken, pig, cattle, sheep, water buffalo, dog, cat, horse genomes, and any other species that you can imagine. We have the fortunate conjunction of having the UK supercomputer less than half a kilometre from us with a high-capacity pipe to transfer the massive amount of data between our machines and the storage facility. We have about 10 times the storage capacity of BGI, so we can actually deal with the data. But when you have all that data, you still need to think about how do we look at a genome. Now, most of the livestock genomes have been published as drafts, and as drafts they're basically, to use the Scotticism, shite. So there are lots of disconnected pieces of DNA are not contiguous. And so to improve the genomes, we have invested in the PacBio long-read technologies, as have other groups in the US. So we're improving the join-up, if you like, of all of the genomes. With tens of thousands of sequences, we can now look at essentially a completely new generation of de novo assemblies using the scaffolds of PacBio. And increasingly, we have to think about metagenomes, because if any two people in this room probably differ by approximately 100 genes that one of you has and the other one doesn't, major copy number variations, etc. And up to 10% of the genome of any large mammal is involved in copy number variation. So when we talk about the human genome, we're talking about Jim Watson's or what's that other guy? Ventnor, yeah. When in fact they are not necessarily typical. In fact, in the case of James Watson, I'm sure he's not. This is being videoed, isn't it? Oh, good. So we increasingly need to look at different ways of looking at genomes, of looking at variation within a species, of looking at blocks, and that requires completely new ways of looking compared to the sort of conventional ensemble and NCBI that we're used to looking at. So when we've got all the sequence, the next stage is to interpret sequence to consequence. How do we go from the DNA sequence into phenotypes that we're interested in? And along the way, we need to go through the epigenome, the transcriptome, the proteome, the metabolome, and understand how all of those things are connected. But at the very first stage, I've been particularly interested in this transition from the genome to the transcriptome, and I've been interested in transcriptional regulation for a very long time. So aside from genome sequences, the livestock genomes are really poorly annotated. If we were farming mice, we would be doing relatively well. But even the mouse genome is relatively poorly annotated compared to humans. And you can tell this by looking at the knowledge of transcriptional variation and the numbers of transcripts produced from each individual locus. In humans, the average is 4.4 that have been well annotated. In mice, it's 3. In the livestock genomes, we're really nowhere near the full annotation of the variant transcripts that can be made from each locus. If we look at the state of functional annotation, in this case the mouse is probably the best annotated genome in terms of function, because we can mutagenize individual mouse genes and look at what happens in an intact mouse. We can look at all of the developmental abnormalities, etc. And there are, according to JAX, there are 24,830 mouse protein coding genes. They've given some kind of annotation to almost all of them. However, only about 2/3 have experimentally derived functional annotations. About 80% are in homology classes, a significantly lower percentage in homology classes with human genes. And as we go down, we find that the ability to translate the information from mouse to other species declines. So the other thing, that's protein coding genes, but the more important part is the non-protein coding genes. At the risk of setting another rat running, if you like, there is a large amount of junk in the genome. Trust me. Most of it is not functional. I think conservation is a reasonable predictor of function, but there is also a considerable gain and loss of functional non-coding DNA between species. And so to understand exactly what's the difference between a cow and a human, it's mainly all the same parts. but they're assembled in a different way, in a different order, and in order to understand that, we need to know the control network. Even within species, within humans, the DNA variants within enhancers and promoters are much more likely to be associated with phenotype. So if we look at the GWAS studies and we look in the ENCODE data and we look only at SNPs that are present in enhancers and promoters, we get a massive increase in sensitivity of detection of genetic associations. So most of the functional variation that underlies phenotypic variation and disease in humans is not in coding sequences. So we need to understand and identify the non-coding sequences and how they work. So in the animal space, there are some names familiar here. I see Brian in the audience. Steve Moore is here as well. The Functional Annotation of Animal Genomes Project, or FANG, I think they came up with the acronym first, as you do, is a coordinated international effort to start to identify the non-coding elements of livestock genomes. It's also, as I'll come back to, really starting to impact on human genetics because, in fact, humans are a great model for livestock. Livestock are a considerably better model for humans than mice are. So I've been involved in genome annotation and transcriptomic annotation since before I left here with the FANTOM consortium based in Japan. And the most recent project from the FANTOM consortium was a genome-scale analysis of transcription using a technology called CAGE. This is cap analysis of gene expression, and basically what it is systematically sequencing the 5' ends of messenger RNAs. Instead of profiling by sequencing the whole message, which is an awful waste of effort because the vast majority of transcription is directed towards abundant transcripts. Approximately 50% of transcription output comes from the top 1% of expressed genes. So if you want to do a lot of sequencing of beta-actin, then you can do whole RNA seq. If you want to get better bang for your buck, then you sequence only the 5' ends. So Riken started with the Helicos, now defunct sequencing system, and have moved, obviously, more recently into the Illumina platform for sequencing 5' ends. This project was published in the last 2 years, and they created a website called Zenbu to visualize all of this data from humans and from mice. Zenbu is Japanese for everything, so there was obviously a massive scale to this project. In fact, in humans, we I think at last count had done CAGE libraries of 2,200 different tissues and cells in different states of activation, etc. So the website is a rather nice one for looking at transcriptional annotation. It shows you a conventional genome viewer, and then down the bottom you can look at the histogram of the expression of the particular 5' ends, and because CAGE tags can be mapped to the genome, you not only get the expression but exactly where it came from. So the viewer enables you to highlight that site, and then it picks up exactly where those transcripts came from. So in the case of SERPINA1 or alpha-1 antitrypsin, we can see that there are actually 2 different lengths of transcripts that have been annotated in full-length messenger RNAs. They are beautifully supported by alternate 5' ends from CAGE, and the upstream one is specific to myeloid cells, monocytes, neutrophils, eosinophils, and basophils. And this one, if we moved the cursor and highlighted this, would be specific only to liver. Now that means when we talk about enhancers, we need to think about how upstream elements interact specifically with one promoter versus another. Alpha-1 antitrypsin is the little league in terms of alternate promoters. One of my other favourite genes is microphthalmia transcription factor, which is involved in lineage-specific differentiation in many different cell types, It interacts with many different transcription factors and tends to have a different partner in different cell types. For example, in melanocytes, it interacts with SOX family members. In macrophages, it interacts with PU1. In microphthalmia, has 7 different promoters. It has a melanocyte-specific promoter. It has a major mast cell and macrophage-specific promoter. It has a kidney-specific promoter, a generic one that is expressed in heart, kidney, and liver, and the one that we knew about most of these, but what we didn't know was retinal pigment epithelium is a specific promoter, and that promoter is mutated in certain human individuals who have depigmented eyes. And so that specific element is required for expression of this gene in the eye. Now, the other thing we discovered with Cage is, very early on, is our textbook view of promoters is wrong. Our textbook would say there's a Tartar box, there's an initiator, transcription initiates, single peak 30 bases downstream of the Tartar box. That is a very exceptional situation. In most cases, if you look at the proximal promoter region, you see clusters of transcription initiation scattered over a broad region. And if you look at different cell types, even if they use them all, they will have preferences for, in this case, smooth muscle using this galactosyl transferase, prefers this distal position, which is in fact a Tartar box, whereas the T cells and monocytes prefer this position, but they will in fact also use the Tartar box. So they make many, many different forms of the 5' end of the transcript. Now, the other way that the Cage data enabled us to look at genes is to look at tissue specificity. Because we sampled every tissue, every cell type imaginable, we could look at the relative tissue specificity. And there's a simple way of looking at that, and that is to look at the ratio between the median of expression and the maximum of expression. If that ratio is very high, it means that there's a subset of cell types or tissues that express the gene and others that don't. And that instantly classifies genes into 2 classes. These ones which are not very tissue-specific at all, the archetypes, of course, being beta-actin, and interestingly, the most highly expressed gene in the human genome, MALAT1, which is actually a non-coding RNA. And I don't think anybody knows what it does. And on the other hand, you have albumin, GFAP, the astrocyte marker, which are very highly expressed in individual cell types. And you can see that over here, looking at the individual profiles and the spread of expression, things like P53 and beta-actin, basically widely expressed, and things like albumin, this is a logarithmic scale, very highly expressed in only a subset of cells. So the other thing with this transcriptional profiling is that when we profile with Cage, and so many genes have alternate promoters, we have 180,000 data points for every cell type that we're looking at. So this is much greater penetrance than microarrays. And it has a much bigger linear range. And so we can look at the relationship between different cell types based on their gene expression profiles. As simply what we call a sample-to-sample correlation matrix. This uses a program which, if you want to look at it, is extremely useful for looking at relationships. It's called BioLayout Express 3D. Doesn't matter what the input is. If you have a matrix where you have relationships between any 2 nodes, you can display it in 3 dimensions. It's actually run off a graphics processor. You can wander through, you can click on your favorite gene and see its neighbors. And you can, I get motion sick doing it, so I don't do it. But in any case, not surprisingly, we have lots of macrophages, B cells, T cells, etc. And as you would expect, their profiles cluster together. That's what you would hope. But what we didn't anticipate, and actually held up publication of this paper, is that most of the cell lines cluster together regardless of what they say they are on the tin. And most of the primary cells of epithelial and mesenchyme origin, fibroblasts, epithelial cells, etc., also cluster together, regardless of what they say on the tin. And the reason why this is the case, we can actually tell why they're clustering together, is that the primary phenotype of a tumour cell line is that it is a tumour cell line. It has all kinds of aberrations of biology and physiology and metabolism that say, I am a tumour cell. The fact that in fact, there's one exception to this, which is multiply aneuploid, which is HepG2. But almost all tumour cell lines are much more similar to each other than they are different. These guys are a bit more of a worry because many of us work on primary fibroblasts, primary epithelial cells, thinking that they are representative of the tissues that they derive from, and they aren't. They adapt to cell culture, to deal with cell culture medium, and they switch off all of the differentiated keratins that are normally a feature of their in vivo biology, and they switch on all of the wound keratins that they would normally express if they were in a bad way. So you can't tell an awful lot from them. Nevertheless, they do have some residual functions of the tissues from which they were derived, and what drives their unique profiles is not surprisingly transcription factors. So if we look at a transcriptional network of only the 2,500 transcription factors in the genome, and we spread them out using, again, this, in this case, gene-to-gene correlation matrix, we can see that the transcription factors essentially separate all of the different cell types into different clusters. So if you know the transcription factor profile of a cell, you know pretty much everything else about it. So the other thing that this profiling does on this kind of scale is what I call guilt by association. There are an awful lot of genes in the genome for which there is no functional annotation, as I said, going past. And the expression profiles turned up some rather surprising ones. And one of my favourites is the informatively named C9orf72. This is famous because it's the major gene associated with motor neuron disease. There is an increasing literature suggesting that both motor neuron disease and other neurodegenerative diseases can have a causal origin in macrophage biology. So my favourite receptor, the CSF1 receptor, is mutated in a disease called hereditary diffuse leukoencephalopathy with axonal spheroids, or HDLS to its friends. But I think people were rather surprised to find that the motor neuron disease-associated gene is in fact a beautifully macrophage-specific gene. So exactly how does that link? So turning to promoter architecture, I mentioned in passing that most promoters do not have a Tata box, and so how do they know where to start? So in that case, we are able to classify promoters into two classes based on the distance between the first transcription start site and the last one, and they fall into two very obvious classes, the sharp ones, which contain Tata boxes, and the broad ones, where the interquartile length is greater than or equal to 11 base pairs. And in fact, about 80% of promoters in the genome fall into this broad class. So then we did what to me is one of the most remarkable indications of the power of the data, and lined up the dominant start site of every broad promoter in the genome, and looked at the sequences either side to see, is there something that says this is to be the major start site? And the simple answer to that is found in looking at the frequency of the so-called WW motif, or A or T, AA, AT, TA, TT, downstream of the transcription start site. And if you look closely, you see a periodic sawtooth with a periodicity of 11.5 base pairs. And you'll know that that's a turn of the DNA helix. And what that does is put A or T on the same side of the DNA helix at periodic spacing, which enables it to bend. And what that does is correlate with, which we also detected in the same data sets, the chromatin structures of, which are these lines here, the chromatin structures associated with poised nucleosomes. So basically, what determines the dominant start site of a broad promoter is the centre of an open piece of DNA with a phased nucleosome sitting downstream of it. Now, to me, to see that signal in a data set of that scale is not much short of miraculous. But the other thing we didn't expect is that CAGE detects the biology of enhancers. And this comes, I need to explain very briefly why this is the case. The RNA that is made in the nucleus can be degraded by a complex called the exosome complex, which eats RNAs from the 3' end, graphically shown this way. So if we look at the conventional transcription of a messenger RNA, we start off head downstream. The exosome complex is in fact sitting on the DNA, bound to the DNA, but it doesn't degrade the messenger RNA coming through. On the other hand, if we look at upstream of messenger RNAs, we actually have a poised active exosome complex which degrades transcripts going in the reverse direction. And in fact, promoters have no intrinsic directionality. The primary determination of whether you make a messenger RNA is whether the exosome complex degrades it. So when we look at the CAGE data, and I didn't emphasize it, you can actually see a little bit of exhaust in the reverse direction on every promoter of these transcripts that have been degraded, that headed the wrong way, got cut off, and got sawtoothed. So CAGE, because it detects the 5' ends, can detect these short transcripts. And when we look at enhancers, we find that there are CAGE tags on both sides of the enhancer. In other words, an enhancer is nothing more than a bidirectional promoter. So CAGE detects these short transcripts derived from bidirectional promoter activity. They're usually about 200 base pairs apart, which of course is basically two sides of a nucleosome. And the interesting thing from my viewpoint is that macrophages have very low expression of these of the exosome, and so we can actually very easily detect enhancer activity. So in my favourite enhancer, the CSF1 receptor, first intronic enhancer, we can detect CAGE tags in both directions. Those CAGE tags are detected only in macrophage libraries. We did chromatin profiling and DNase hypersensitive site analysis for the cells of peripheral blood, monocytes, NK cells, CD4 T cells, and B cells. And the CAGE tags, so these are centered on the enhancers detected with CAGE, and you can see that in every case we see also the tissue-specific signatures as DNase hypersensitivity or chromatin modification, each side of those enhancers detected with CAGE. So CAGE enables us to look at transcriptional regulation, simultaneously looking at the activity of promoters and enhancers in the same cells across time. And of course, many of you will know, and I'll use a photo that I first stole from Jenny Stowe many years ago, that I work on macrophages, and we've of course applied this technology to macrophages by doing a very dense time course of the response of macrophages to LPS, and I'm not going to show influenza. So this is the time course, 15-minute intervals, and then increasing right out to 48 hours. This is just one example of one of the genes we look at, interferon beta-1, which is induced transiently by LPS. It's also evident using this technology that one individual is a much better interferon producer than some of the others. We actually know why that is. There's a polymorphism in the interferon promoter in humans, which means that some of us are much better interferon producers than others. So this is the full profile of the response to LPS and an unconventional heat map, because green seems to me to be much more associated with go than stop. So looking at the time course and clustering sets of genes with common regulation, you can see that one of the earliest responses to LPS is actually very rapid downregulation of a whole set of genes. And we tend to ignore those. We're all much more interested in activation than repression. And then there's this incredible cascade that goes all the way out to 48 hours and is still changing at 48 hours of genes that are induced and repressed, wave after wave after wave of transcriptional activation and transcriptional repression. We tend to think of macrophage activation in terms of making TNF-alpha and the rest is history. Inflammatory cytokines is all there is. But in fact, this is a much more complex transcriptional regulatory phenomenon. As I said, we can also use CAGE to identify the enhancers and their relative transcription relative to the promoters. And I showed you serpin A1 earlier, or alpha-1 antitrypsin, and the upstream promoter. The upstream promoter actually segregates into 4 separate transcriptional start site clusters, all of which are induced by LPS late. And if we look at the activity of the enhancers, which are also visualized on the Recon site, you can see that 4 out of the 6 enhancers associated with this gene are induced transiently by LPS, well ahead of the induction of transcription of the promoter. Transiently. So there is an event that switches this enhancer on, then the job is done and transcription happens. The star of this show is an LPS-inducible gene, DUSP1, which is induced biphasically. There are actually 16 enhancers in the vicinity of this gene. At least 8 of them are induced by LPS. It's a bit noisy because the numbers of tags that we detect are relatively low, but it's clear again that they're induced in advance of the first peak, and in some of them there looks to be a secondary peak of activation associated with the second activation of the promoter. So what we learn from CAGE is that every gene has multiple promoters and even more enhancers. Enhancer activation occurs before promoter activation is transient. And I emphasized earlier that enhancers vary between individuals, and they also vary between species. And one of the ways we've looked at variation between species, when I was here, started a project with Matt and Kate Schroeder and others, looking at differences between mice and humans. And we've continued that comparative biology recently, looking at the different responses of mouse and human to glucocorticoids, which are the major therapeutic agents in anti-inflammatories. And when we look at the relative response of human macrophages and mouse macrophages to glucocorticoids, the overlap of inducible genes is about that. So the vast majority of glucocorticoid-inducible genes in humans are not induced in mice, and vice versa. And so we did ChIP-seq analysis to identify why, and what we found was that DUSP1 is induced in both species, but even in that, we have a glucocorticoid receptor binding site in mouse that is not present in human, and one in human that is in a completely different position in mouse. These genes, so this is a representative of the small subset that are induced in both species, and on the other hand, if we have a whole set of mouse-specific inducible genes like factor 13A, induced in mouse but not human, associated with a glucocorticoid receptor binding site within an upstream enhancer that is not bound in human, and a DORA3A, which is associated with an intragenic enhancer site in humans, which is not bound by glucocorticoid receptors in mice. We can also tell exactly why this is the case, because we can look at the sequences around these set sites that are bound by the glucocorticoid receptor in ChIP-seq, and we get beautiful enrichment for the glucocorticoid receptor recognition sequence in both the mouse-specific and the human-specific bound sites. So Between the species, we gain and lose binding sites for glucocorticoid receptors, and accordingly we gain and lose, with single base substitutions in many cases, the ability to induce the gene in response to glucocorticoids. The interesting thing is that these enhancers that are bound by glucocorticoids are actually conserved globally, so they're regions of conservation, but within those regions of conservation you gain and lose binding sites for specific transcription factors. So returning to animals, if you want to drive yourself crazy, tout est bon dans le cochon is everything is good in the pig. And if you go on the internet and look on YouTube, there's an incredible song that goes with this, which you'll never get out of your mind. So we have spent a lot of work recently annotating the pig genome. A couple of years ago, we did a transcriptional atlas based on microarrays and designed a new microarray platform for pigs. But we've more recently started to ask how much of the data that we have from mouse and human can we translate to pigs from all the FANTOM 5 data. And I won't belabor this, but we mapped all of the human promoters, 180,000 of them, and enhancers to pigs and to mice. And you'll see that of all of the promoters and enhancers, only a relatively small proportion of them map to both species, and a significantly greater proportion of them map to pig and human, but not to mouse. So essentially, the long and short of that is that pigs are rather more human-like than mice are in terms of conservation of promoters and enhancers. Mapping these elements across can give us a preliminary annotation of the likely expression of a gene of unknown function. More locally, of course, I have an interest in specific regulation of certain genes like the CSF1 receptor promoter. CSF1 receptor promoter is an example of a gene that has no Tata box, but also lacks a CPG island. And so it's not a GC-rich promoter, it's a purine-rich promoter. And if we look across species, it's reasonably conserved, but these green and yellow areas are the purine-rich elements, which are not terribly well conserved across species. And so we've actually started doing CAGE on a large scale in pigs, and starting obviously with macrophages, and compared the transcription start sites of human, pig, and mouse, and the relative location of these PU1 purine-rich binding sites, and also the binding sites for the Ewing sarcoma protein. And the PU1 site determines, in essence, where the transcription start site cluster will be, because it actually breaks up a nucleosome. So you can see in mouse, there's a single cluster. In pigs, the two PU1 sites are further upstream in the alignment. And in humans, there are essentially a duplication of those PU1 sites, which means that there are two peaks of transcription initiation. So even though the elements are conserved and the promoter looks conserved, the precise detail of exactly where you start transcription differs between species. So the final thing I'm going to mention is the Sheep Atlas project. In this case, we have started a project looking to improve the annotation of the sheep genome. And in our particular case, we are looking at F1 crosses between a classic terminal crossbreed, the Texel, which is also the reference sheep genome, and, of course, being where we are, the wee Scottish blackface, which is hill-adapted and has all sorts of interesting traits of hardiness, survivability, and disease resistance. And the reason that we've chosen these two breeds is we want them to be genetically as different. Yes, take your eye off the buttocks. I'll go back one. There. Now, concentrate. What we want to do is use the deep RNA seq to look at the relative expression of the two alleles, the transcripts from the two different chromosomes from the parent. There's been very little work done on allelic imbalance, but in theory, if you have two chromosomes in the F1 animal and they have a genetic variant, that alters the binding of a transcription factor on one allele or the other, then with deep RNA seq, if this one is more highly expressed than this one, we'll get more examples of the C-containing sequence than the T-containing sequence, and we'll be able to infer that that C is linked to a variant in the promoter or the enhancer that controls expression. And so we can be able to start to understand transcriptional regulation at a single allelic level. So we do know about imprinting, and so this is one of the examples of severe, if you like, allele-specific expression, the archetype of the whole field, really, the Calipese sheep. So this project was funded by BBSRC and has gone a long way. We've actually got all of the sequence data now. 370 messenger RNA libraries and 72 total RNA libraries. We've also looked at blastocysts, and we have actually about 10,000 RNA samples in the freezer if we can actually find the money to continue sequencing. We've created a preliminary atlas, and it looks like it's going to work rather beautifully to annotate. This is again looking in biolayout express at the co-expression of different genes. So there's a cluster of genes in the GI tract, immune genes, reproductive, heart, blood, and more extreme immune genes. So this is quite useful because the sheep genome is not terribly well or informatively annotated yet. There's lots of things that basically give you this kind of number. But if you see this gene expression profile and you see that all of the genes in this cluster show this kind of average expression profile expressed in gastrointestinal tract, and you have known genes associated with gastrointestinal differentiation, the chances are that these genes have got something to do with gastrointestinal differentiation as a preliminary annotation. And similarly, if you have a cluster that is completely muscle-specific in its expression, and within that cluster you have skeletal muscle alpha-actin, the chances are that these genes are likely to be muscle-specific in their expression. The bigger the cluster that you deal with, the bigger the data set, the more confident you get about the guilt by association. The statistics get better and better that certain genes have particular functions, but also you can start to break these clusters up. And one of the examples we got from the pig gene expression arrays was to recognize that encoded mitochondrial genes are not perfectly correlated with nucleus-encoded mitochondrial genes. And so we also annotated some new mitochondrial genes in the process by association and confirmed them. But that also tells us that there's not perfect regulation of the production of the proteins that are encoded by the mitochondrial genome, coordinated with those of the nucleus. So I'm going to finish up there and just summarize this journey through all creatures great and small. Genomics and genome editing are rapidly changing animal and plant breeding. They will drive another genome revolution. I think the combination of sequencing and editing is the biggest thing in genomics since PCR. Without a doubt, it will revolutionize and change our world. In order to use that technology, we need better genome annotation, and we particularly need functional analysis of the non-coding elements. Exactly what does a variation in an enhancer predict in terms of the regulation of the gene? Gene regulation is complicated. Comparative analysis within and between species can give us novel insights into how transcription regulation is controlled. And finally, transcriptional network analysis can predict likely gene function. So just to give the credits, these are colleagues in Edinburgh, colleagues in the Recon Genome Consortium, and colleagues within my laboratory in Edinburgh who've carried out most of the work. Thanks.